Good afternoon. I'm grateful for the opportunity to share our vision and work with you. My name is Jae Jung-ho of HADD Science, a company dedicated to creating scientific technology. We think the science should add the value to human life. So that's why we call HADD, the hard science, not the hard. Our company develops technology for 3D cell culture and organoid-based stem cell research. As many of you already know, the FDA abolished existing animal test in the new drug licensing requirement called New Approach Method, the NAMS, for reduction of a preclinical trial period and overcoming interspecies differences, also and reduction of animal sacrifice. This means that drug efficacy and safety evaluation will increase, rely on human-derived 3D cellular in vitro system like a spheroid and organoid rather than animal test. This will not only accelerate large-scale drug screening, but also expand into personalized mini organ platforms for the cell therapy and regenerative medicine. To prepare for this shift, researchers and companies worldwide are working to standardize organoid production. However, when we culture organoid in vitro, like here, brain organoid, we often see significantly variability in size or morphology and even its functional state. So the high variability and low uniformity must be solved for large-scale organoid production. So our approach introduced a new way to culture organoid in a floating environment similar with the human body condition. So traditional liquid-based media require constant shaking to prevent organoid from attaching to the plate surface, a process that often increases variability. So in contrast, our material we call HADD gel transform any liquid medium into the gel-type medium simply by adding it into the solution medium. So you can see the organoid can be floating state during their culture. So this floating state to remain suspended uniformly in large number of enable consistent scalable production of organoids. So when we survey 3D culture users, they consistently mentioned several challenges, difficulty in handling, high cost, and concern about toxicity. The most widely used ECM gel is Metrigel. It's powerful, but it contains carcinogenic factors, and its viscosity changes with temperature and making experiment unpredictable. So we built HADD gel differently. Its viscosity stay stable with the temperature changing. It avoids carcinogenic component because it only contains deoxyribonucleic acid, DNA. This is a safe biomaterial, and it's available at a price point around $300, making it an accessible and reliable alternatives. So to deeply understanding organoids, we needed to see them alive in motion in 3D. So we developed also live gel too. This material that stabilizes organoid for long-term imaging and enhance their fluorescence contrast. So it allows researchers to watching living organoid growing, express fluorescence tagged signal in response to drug with the massive number of organoid at the same time. Through our advanced biomaterial, we are building a comprehensive platform for 3D organoid, spheroid, and tissue-based cellular system. Our direct market, the cell media, cell culture material, and 3D cell culture market, they are quite rapidly expanding. And then also broader cell culture market is growing even faster, driven by drug development and regenerative medicine. Since our company founding, we have begun generating revenue, and we believe global expansion will significantly accelerate our growth in here. We actively engaged with researchers at many conferences and publish papers and patents and collaborate with the organoid CRO company and equipment companies. We have recently signed a distribution agreement with a regional supplier in Korea and also in US, partner with the 3D culture plate manufacturer named 3D Bioprinting. Yeah. And one of our primary goals during this US visit is to establish partnership with the global distributors and also research technological company. So far, looking ahead, we plan to collaborate with the major pharmaceutical companies, CRO, and eventually hospital conducting cell-based therapy. There are, yes, our core member of HADD, this team. These two of us, them is come here together with me. The Dr. Ari, team leader in R&D section, and Harry No, product manager. We are also fortunate to receive strong support from Korea government agency in focus of NAMS development, organoid standardization, and advanced biomaterial research, and also startup acceleration part. So our history is maybe short, but our story is already long, and now we wanna go bigger from here. So not only in developing the biomaterials and cell culture technology, but also in shaping the future of the brain organoid modulation and cell therapy with organoid transplantation. Then you can come and join HADD Science. Let's work together to create technology to truly add the value to human life with HADD. Thank you.
Could you please speak about the competitive landscape?
The competitive landscape?
Who are the competitors in this space?
Competitive. Maybe the Matrigel company. You know, the Matrigel, they are Corning company. They supply the ECM protein-based gel, but this is not the media. We developed the media, but ECM gel material is only short-term growing section we use, the researcher use, but for the long-term organoid culture, like 1 month, 3 months, the organoid need the media, liquid media. So we suggest our material to growing the plotting system.
And the technology, can that be used for variety of cell types?
Yes. variety of organoid, like a lung organoid, heart organoid. The people co-occurred, they used it.
Okay.
Yes.
Thank you.
Thank you so much.